This is TWIS. This Week in Science, episode number 742, recorded on Wednesday, October 9th, 2019, Science Nobelity. I'm Dr. Kiki, and tonight we will fill your head with resistance, false memories, and mold pigs. But first,
TWIS is supported by listeners like you. We thank you for your support. We really couldn't do it without you.
Disclaimer, disclaimer, disclaimer. The following program has persisted out of a love of science, a belief that the discovery of scientific knowledge, application of that knowledge in the world, and a constant effort and dedication to learn more make the world a better place to live. Through science, we accomplish the goals humanity sets for itself. But science is in trouble. There are powerful people who simply do not want scientists to speak publicly, want to prevent them from sharing their knowledge. Not that this is anything new, for science has always been in trouble this way. Religion was an early and persistent threat, demanding through violent retribution that no progress be made in science that did not capitulate to the prevailing dogma. Currently, the threat is political, with redacted reports, researcher retaliation, and silencing of results. It's happening at an increasingly inhibiting pace. Maybe what we need now is a new legal framework, a constitutional amendment for science, a protection for the funding and findings of science, one that separates once and for all the political interests of politicians and their donors from the work of scientists. Separation of science and state, Freedom of Publication Act. One that once and for all prevents the willful disregard of scientific findings by political appointees and instead puts the knowledge in the hands of This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
and a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to thrill you with sights, well, not sights, sounds of science. Stories to fill that mental visual space in your brains with curiosity. All right, I have stories. Well, you know, I'm going to be talking about the Nobel Prizes tonight. That's one thing for sure. I also have stories about false memories and zebra stripes.
Again,
Bringing it
Can't
back.
wait.
Yeah, you're going to love this one. Justin, what did you bring?
Oh my gosh, there was so much to choose from. I've got antibiotic resistant chickens, tropical Mexican miracle cures, human regeneration, mold pigs, as you mentioned, and a 400,000-year-old camel.
I think the mold pigs has won for me tonight. That's the one I have curiosity about. I gotta know. And Blair, what is in the animal corner?
oh my goodness. Well, I am so excited that my plane landed just in time for me to run to the show tonight. And I have brought from various hotels and airports some science news.
Diseases.
And
No.
that is, well, luckily that's the beauty of the teleconferencing, I suppose. But I have brought robots saving animals, TV saving animals, and bird pee.
Bird pee.
Oh,
Yeah.
The birds don't pee. But let's talk about
We're going to
that
talk
later.
about it. Yeah.
All right.
wait,
We'll
what?
We'll talk about
Okay,
what
now I'm
passes
really
through
curious.
that cloaca.
Yeah.
Yes. As we jump into the show, I would love to remind you all that you can subscribe to the This Week in Science podcast everywhere podcasts are found. Just look for This Week in Science. You can also go to TWIS.org for information about the show and to find out about the 2020 TWIS Blair's Animal Corner calendar. You can pre-order that now. But it is now time for Nobelity. So as I get started here talking about the Nobel Prizes, once again, last year it was like this exciting year because there were 2 women in the sciences who got prizes in the Nobel Prizes, and everyone was like, yes, it's a step in the right direction, more women getting awards. And this year it is a bunch of men again, which is great. They're all very well deserving, and these are amazing discoveries that deserve recognition for what they have done. But, it would be nice to see a few more women on the list. We're going to we've said it before, as the Nobel Prizes continue and as they look at things differently and as more women are dominant in their fields in the sciences, as they have been for the last couple of decades and growing, you will see more and more women receive Nobel Prizes. It's going to happen. There is traction. The first prize for the Nobels that I want to announce, if you haven't read already, the Chemistry Prize. It has been presented to 3 researchers, John B. Goodenough, M. Stanley Whittingham, and Akira Yoshino, for developing lithium-ion batteries. The batteries that we have in our phones, the batteries that catch on fire on planes, the batteries that are in our laptops, the batteries that allow our rechargeable portable lives to take place these days. These gentlemen were responsible for developing lithium-ion batteries, not all together. They didn't all collaborate. This award is given to these researchers for the sequence of iterations that each of them made on the previous researchers' work. Stanley Whittingham, that during the oil crisis in the 1970s laid the foundation for the lithium-ion battery technology. He was researching superconductors, discovered an energy-rich material that he used to create an innovative cathode in a lithium battery. This was titanium disulfide that could intercalate or house lithium ions. And then Goodenough predicted that cathode would have greater potential if it was using a metal oxide instead of a metal sulfide. So he changed the chemistry a bit. And then Akira Yoshino created the first commercially viable battery in 1985. These things have been around for a long time, and they don't look like they're going anywhere anytime soon.
It's an interesting way that they've done it on this award. It's rare that you see a physical invention being part of a Nobel, and it's maybe for largely the reason that you were mentioning is that the iteration of an invention has so many portions to it, and in previous discoveries and previous accelerations of knowledge that are required to get to the next step. And there's a perfect also example of the handoff of shared knowledge where, oh, look, I found this really cool thing. Don't know what to do with it, but it's really cool. And then somebody else picks up like, oh, I've been looking for something that does exactly that. I just couldn't conceive of it. And then somebody's like, oh, well, let's make it. That's actually
But I mean, that's like, we talk about CRISPR all the time, and that's exactly what has happened with CRISPR. It's people iterating on the technology, the finding that bacteria have this mechanism, and then how can we use this, and what can we do with this? And it's still being iterated on, and it's an amazing tool to science now. And
none of this takes place if this knowledge isn't shared. Right. Is the big thing. If this is siloed and some patented or not, someplace where somebody's trying to develop, and maybe it never turns into a battery. This cathode initially gets discovered.
For the Nobel Prize in Physics for 2019, this was awarded 1/2 to James Peebles for theoretical discoveries in physical cosmology. Basically, the Big Bang. He's got some. He did a lot of work and the Big Bang. And the other half went to Michael Mayer and Didier Queloz for the discovery of an exoplanet orbiting a solar-type star. So this is the first Nobel Prize given for exoplanets, which is, in itself pretty impressive. I was looking up some information about who had discovered the first exoplanet. The first confirmed discovery of an exoplanet was January 1992. Radio astronomers Alexander Wolksan and Dale Frail announced 2 planets that they discovered around a pulsar. But it was Michael Mayer and Didier Queloz of the University of Geneva who they announced the first definitive detection of an exoplanet orbiting a main sequence star. And the technique that they used, the technology that they used, really laid the groundwork for what is now the modern era of exoplanetary discovery. The high resolution spectroscopy that they used was able to further advances as it was used more and more. But it's interesting that the people who got the award were not the first discoverers of an exoplanet. So maybe sometimes if you get there first, it's not even enough.
Interesting.
And we found so many of them too that we've become very blasé. Like,
Absolutely.
okay, is it one? We only care. Can we live on it? It's the gravity's too much. We've gotten very specific, very snobbish about talking about exoplanets.
It's really now. It's so hard to even contemplate the idea that exoplanets at one point were just an idea, that we didn't know that they existed. I mean, I feel like, of course there's exoplanets around other stars. Of course there are exoplanets. We're finding them all over the place.
That's what I was thinking. I don't understand why that's if we knew that stars were suns, basically, right? Like, seems.
But you have to, it's science. We must confirm.
Yeah.
And
for sure.
if you go, you don't have to go back that far. Maybe it's 100 years. When we knew of one other galaxy,
right?
Mm-hmm.
We now have photographs of billions of them. It's really science.
It's pretty hard to break through those barriers where we admit that we're not special. I feel like so. Even if we said, okay, there are other suns, but they don't have planets. I mean, that's just us. Right.
I found it a complete relief.
Just us.
Like, thank goodness. Oh, thank goodness. This
for
really
you,
isn't
but I feel like for many others.
When I look around, I'm like, thank goodness. Mm-hmm.
Yep. Well, James Peebles, we have to say thank goodness that he came up with the theoretical framework for the Big Bang model, and his theoretical tools and calculations allowed an interpretation of traces of radiation in the universe to allow us to discover what is in our universe. That his results showed a universe in which just 5% of its content is known. The matter which constitutes stars, planets, and trees, all of that baryonic visible matter. The rest is unknown dark matter, and still is a mystery. Don't you forget it, people. We still don't know what the rest is made of, even though people sound very convincing in books and on podcasts. And finally, the award for physiology or medicine has been awarded to William Kalin Jr., Sir Peter Ratcliffe, and Greg L. Semenza for discoveries of how cells sense and adapt to oxygen availability. And they identified molecular machinery regulating the activity of genes inside the cell's metabolism to varying levels of oxygen. So it lets us understand what they did discovering this oxygen sensing machinery. Allows us to really be able to understand how cancer cells work in their anaerobic or aerobic situations. They help us understand how changes in oxygen availability could influence growth of cellular populations or of just living organisms. The researchers discovered a gene, the EPO gene. They studied it and how it gets regulated and mediates the response to hypoxia. Studied oxygen-dependent regulation of this gene. They found this mechanism was present in virtually all tissues, all over the body, so everywhere. How does our body deal with it? Yeah. Oxygen sensing. This is one of the things. Oxygen is important to life, and so this mechanism is inherently important. Moving on from the Nobels, there are more awards to be given next year, and let's see if some more ladies make the cut. We will be hoping for that. In the meantime, funding of scientists is something that's very important to researchers actually being able to do groundbreaking work and actually make these impacts that end up changing our sense of the universe around us. We've known for a long time that Black scientists are less likely to receive funding from the National Institutes of Health. National Institutes of Health are one of the major funders of medical and health research in the United States. And a study was done looking at some 157,000 grant application proposals submitted between 2011 and 2015 for what are called the R01 grants. And these R01 grants are very large grants that form the ability of labs to be able to do the work that they do. They really give that, they let labs run and exist. The researchers placed each proposal into one of 150 topic areas and examined factors that could influence the final outcome, finding that 3 contribute to the gap that in funding that is actually seen. This is called the Ginther gap. They found that the score, whether or not it's scored, more than half of all proposals don't get any score at all. What score it receives, which this all kind of makes sense. The higher the score, the better it does, the more likely it is to get funding. And the factor that had the highest contribution was topic choice. So what were the researchers applying to actually study? Yeah, and so the question now is how are the perceptions or the priorities of the Black research community, and how do they differ, or how do they compare to the priorities of NIH and the NIH proposal analyzing committees? Because something is not lining up. And this study doesn't actually get into that and does not answer that question,
Right.
but it raises that question for
Right.
sure now.
So in my field, in the field of environmental education or informal education, depending on what bucket you're looking at, there is this large conversation happening right now around diversity, equity, and inclusion, and
Yes.
specifically in trying to get people that aren't just white women teaching at science museums and zoos and aquariums, because that's everybody who's doing it pretty much, right?
Yep.
And why is that? And how much of it is based on the fact that that's just who's currently doing it, so other people don't see themselves in that line of work? Or what a lot of places are doing now is they're actually bringing in outside consultants to look at their hiring process, to look at their posting process, and specifically to look at job descriptions. And it
Yeah.
turns out that there's a lot of stuff that people put in job descriptions that can alienate entire groups of people, that we don't even realize. And so I see, lots of talk in the chat room saying, I can't believe this is an issue. I don't think that necessarily it's always intentional. And I think that a lot of times there are these kind of grandfathered in expectations that we don't realize are causing these larger systematic effects that are keeping us from being a better reflection of society as a whole. And so I think the more I hear about stuff like this, I think it brings us that further to light, that we need to bring somebody into our systems that are not representative and really take an outside look and see what biases we're accidentally bringing into our hiring and our, scholarship awards and all these sorts of things.
Absolutely. And this is definitely an issue here. And one idea that's being bandied about is that there are more studies related to actually applying our existing knowledge about health and medicine to communities and the systems that are in place to improve systems that we have, whereas the NIH focus is potentially more so on basic science and making new discoveries, as opposed to taking the knowledge that we already have to improve the way that we do things. And this is a potential explanation, but it, as of yet, doesn't have any evidentiary weight behind it. But the researchers from this group say that at this point in time, the reviewers are not consciously looking at these things in a particular way, and that there's maybe just some differences in the system and the way the system is set up that need to be addressed in the exact way that you were talking about, Blair.
Yeah. So one of the other things that's a factor in this often, and we covered a study about this 10 years ago or something, is that there's also a tribalism in how you hear about jobs. And so if your friend circle is not very diverse, the jobs you are most likely to hear about are jobs that people in your friend circle with their lack of diversity are more likely to be in, sort of a thing. So you will end up with a lot of white males in Silicon Valley at a company that has hired mostly white males.
Right.
But if there is a more diverse culture there, there's more people who will hear about jobs and know to apply for things and this sort of thing. Which kind of makes sense to maybe it's a white woman circle of tribe got those jobs early and is referring other women to do them. Okay. But when you get into these are grant funding statements or requests that are coming through in just document form, you can still apply this, because who knows to apply for these in the first place or what the application process is. There is still going to be a tribe of people who know to do applications, but in the percentage that is getting, then you're starting to also talk about what do these specific tribes care about? What are the topics that are being discussed? So if and we don't know that there is, because I guess they haven't talked about specifically in this, if there is a specific different subset of issues that are being of concern from the community of color, as it were, in the scientific field, if they're actually attacking different issues or what that's about. But that would be an interesting dive to find out if there really is a philosophical approach that is different in some way.
Or just not represented. It's just, not represented yet. So it's definitely an area across the board that needs improvement, to see greater representation and to see, greater diversity in who gets funded, ideas that are getting funded, and let's do the best we can. And this research is great for pushing that forward. And hopefully there will be more studies that actually delve into these kinds of questions more deeply. This is This Week in Science. Hey, Justin, you want to tell me a story?
Is it time for me to say stuff? Okay.
Yes, do it.
So humans have been overusing antibiotics quite some time now, to the point that antibiotics are becoming somewhat resistant to our efforts. It turns out, though, we are not alone in this. Animals also have been overusing antibiotics at an alarming rate, it turns out. So this is nearly 1,000 publications and unpublished veterinary reports from around the world were used to create a map of antimicrobial resistance in low to middle income countries, livestock. They focused on bacteria, E. coli, Campylobacter, Salmonella, Staphylococcus, all of which cause serious diseases in both animals and humans. Since 2000, from the results of the study, since 2000, meat production has accelerated by more than 60% in Africa and Asia, by 40% in South America, as countries in those continents get a taste for meat in a higher degree. More than half of the world's chickens and pigs, just a fun fact, are in Asia currently. The study found that between 2000 and 2018, the proportion of antibiotics showing rates of resistance above 50% in developing countries increased in chickens from 15% to 41%, and in pigs from 13 to 34%, meaning that the percentage of livestock populations that are becoming majority resistant to antibiotics is increasing about threefold in 2 decades. So, researchers found that antibiotic resistance in livestock was most widespread in China, India, and Brazil, or China and India, with Brazil and Kenya emerging as the follow-ups. And meat production accounts for this. I guess I should have known. Makes sense. Meat production accounts for 73% of global antibiotic use.
Yeah.
And this is what sort of made these large-scale farming operations even possible, because in the olden times, you would never house as many pigs, chickens, cows, what have you, together, because if one or two of them got sick, all of them would get sick, and then you would have a bunch of sick, dead animals on your hands. So one of the things they're pointing out, and this is the tough ask, or it's in any regard, is that researchers are suggesting that developing nations should take action to restrict the use of human antibiotics in farm animals, and that the affluent nations should support a transition to sustainable farming. So this is a tough ask in 2 ways. It's asking nations who aren't profiting from these industries to pay. Come up with a fund and international biosecurity funds to address the issue. But then it's also asking developing nations who are already not in great financial shape, perhaps, in terms of the money they can afford to put into food and raise food for their population, to take on an additional expense. So chances are nothing will happen, right? But this is sort of an alarming state of things for the rate of this increase, and the idea that it's unregulated too. Like we've only just really gotten to the point where we understand how much we need to regulate antibiotics in developing nations, and now to see that that all of our efforts locally are likely to be undone by the proliferation of these farming techniques globally. It's sort of like having to deal with our younger selves in a weird way. You know, where we
Right,
were,
exactly,
where we were 20
were.
something years ago, that we're like, okay, that was a bad idea. Now we've got the solution. Oh, but our old selves are still here right alongside of us, causing the problem. So
And
it makes it difficult.
we should be addressing that, and we've figured this out, let us help you do it in a different way. The issue is a lot of agriculture in North America is still using massive amounts of antibiotics. Maybe it's more regulated than it was at one point, but I don't think so. But
it also sort of indicates that, because these things can occasionally swap from animal to human, and if we have all of the efforts of preventing antibiotics from becoming ineffective in our human population, could easily be outdone by creating highly resistant strains in animals that then move over. So
yeah, and there's these antibiotic.
That's written all over it.
Yeah, it absolutely does. But even beyond that, I was just looking. I remembered a study that I saw as I was researching for the show this week. They're finding that the animals who get the antibiotics, like cows, when they defecate, so it has an effect on the soil. And so researchers at Colorado State University and University of Idaho discovered that there were changes in carbon allocation and carbon use efficiency. The study is titled Prolonged Exposure to Manure from Livestock-Administered Antibiotics Decreases Ecosystem Carbon Use Efficiency and Alters Nitrogen Cycling. So the antibiotics have a trickle-down effect on the ecosystems these animals live in as well. And so there are multiple layers of of bad here.
Yeah, there's some analogy of something running downhill.
Yep.
That is. So basically what as we are losing antibiotics solutions to things, this is of course the predicted doom of the world over the next 30 years. However,
Yay!
there is good news. Thanks to the tropical forests of Mexico coming to the rescue, a new hope for antibiotics has emerged, and it's has some unique properties that could make it actually pretty attractive in agriculture as well. So it's, I don't know, I can't pronounce this. Phasalicin is with a pH for the it is the compound.
What are you?
Phasalicin? I don't know.
Phasalicin?
Yeah, it could be lysine. It's going to be a household word in 10 years.
Yeah.
It'll be a household word in 10 years. We'll figure it out then. Antibiotic resistance.
Z for short. There you go.
This is Konstantin Severinov, a molecular biologist and biochemist at Rutgers University, New Brunswick. Antibiotic resistance is a huge problem in both medicine and agriculture, and continuing searches for new antibiotics are very important as they may provide leads for the future antibacterial agents. This compound is found in root nodules of wild beans in tropical forests of Mexico. It's unusual because it is an antibiotic that is produced by a symbiotic soil bacteria that fixes nitrogen for the plants and helps them keep microbes away. So it's doing two tasks. So it's fixing nitrogen and keeping unbeneficial microbes away from the roots of the bean. It has this really unique action though, in that it actually goes in to enters the bacteria that's trying to attack the root, binds to the ribosomes, and basically messes its ability to synthesize proteins. So this is really a shutdown operation in a completely different way than most of our antibiotics have been working. It essentially shuts down the machinery inside of the microbe. So, and in this also, it looks like it can be useful and to the plant in terms of growing and proliferating and making a healthier crop. So this is a 1-2. This is something that could actually be used in agriculture as a nitrogen fixer. That could also be a natural probiotic for plants that you're growing. And if this peptide can be reapplied, it can be used as an antibiotic, perhaps in humans and cows and pigs and anything else. And it's one of those things that you wonder how well there would be a defense for it. If this can bind to a ribosome, this is attacking before a defense system with the machinery that the defense system might generate new proteins, novel proteins, something other than what was affecting that bacteria in the first place as the overcoming of the antibiotic. But if you're shutting down the machinery before it has a chance to react, that's a really powerful way of going about it.
Yeah, that is really interesting. I mean, it's kind of like it's getting in there and gumming up the works. So you have the RNA coming off the DNA, and then that RNA is going to go over to the ribosome to get turned into a protein. But then this thing gets in there and says, nope, stop. Just stop right now. That's not going to happen. And so you don't get any translation. You don't get any of that actual production of the gene into a protein to do what it needs to do. And so this seems like a mechanism that could be taken and used like for not just one purpose or one species, but maybe multiple. If you can get it in to attack and stop the translation, it could potentially be, who knows? It could be a really good biocontrol agent.
It could also accidentally wipe out your entire microbiome, right?
Okay. What?
It sounds like it's something that if you found a way to make it kind of more universally effective, it could accidentally just kind of like, especially since if you're keeping it from being able to replicate, then that means that it, in theory, would avoid resistance. Right.
Yes, but the thing to keep in mind is this is a peptide that is created by a bacteria acting on a root So this is not a living thing that is going to be proliferating. Of course, will be in a set amount, right? I think I might have some pretty interesting research applications too. That would be pretty fun. Being able to go and shut down ribosomes for periods of time might be a very useful tool.
And I mean, it's also possible that, with everything we've learned about the microbiome, for example, you could potentially just want to nuke your entire microbiome and start over.
No.
So you could do that.
Don't do that.
I mean, if you have a really bad gut problem, you could just pretty much.
You're right. Gut, absolutely. Right? Yeah, there actually are.
Short of drinking bleach, this would be much more effective.
Yeah, but I mean, then we're saying this is how people have addressed before is actually taking antibiotics to sort of do the antibiotic cleanse. But again, running out of antibiotics, here's a new potential that can maybe save the planet.
Fingers crossed. So you gave us some bad news and then some good news, and
I try to balance it.
Try to balance it. It's good, and then now it's time for Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels, there's Animal Corner.
What you got, Blair?
Oh my goodness. Well, I'm excited. I have all good news this week in the Animal Corner. I may have been kind of inspired by the past 10 days that I spent talking to people researching out in the wild, but I have a couple of really cool stories about animal research that's going on. So the first one is actually about using drones for conservation. And of course, we've talked in the past about how drones are being used to monitor rhinos in the wild so that they can be protected and we make sure that people aren't poaching them. And drones have been used to follow animal movement and migration. The latest use for a drone in the wild is to weigh whales. Weighing whales with a drone. Yes.
How do you do that?
What?
Well, I'm going to go ahead and put air quotes around the word weigh. Let me explain what's actually happening here. So this is researchers from Aarhus Institute of Advanced Studies in Denmark. And Woods Hole Oceanographic Institute, WHOI, in the US. And they have found a way to accurately estimate the weight of free-living whales using only aerial images taken by drone. So this is an estimation of weight. They are not actually weighing the whales with a drone. I'm not exactly sure how that would work. I'm imagining some sort of amphibious flying drone with a sling, but no, that's not what this is. It's just a picture taking device. So they can measure the body length, width, and height of free-living southern right whales. And they took these photographs. They measured these different metrics on the whales. They were able to find a model that accurately calculated body volume and mass of the whales using those images. So obviously the way that we've only measured whale weight in the past has been by weighing dead or stranded individuals on beaches, which is a hard way to measure that because at least in the case of the dead beached whales, they're bloated. Decay has set in. Their weight to measurements are not exactly what they should be for a living whale. So it's not a good place to start. So this is a really big kind of jump ahead in that. They think that also they'll be able to track individuals and watch their dimensions grow over time so that they can have better understandings of how whales grow and how they gain their weight and what a healthy whale looks like, so that when we're looking at conservation concerns with wild whales, we can have a good idea of what it's supposed to look like and kind of go from there. So this is all very new.
It's very new. I mean, the ability to be able to have this kind of monitoring, we've never had this ability before. This is really interesting.
Yeah, so they're saying that they can now look at the growth of known aged individuals, calculate their body mass increase over time. That's kind of what I was talking about. But then extrapolate that into energy requirements for growth and take that information, look at daily energy requirements for whales, and calculate how much prey they need to consume. So now if you're thinking about ecosystem needs, you can actually see, okay, if this fishery is declining, this is what it might mean. Or a lot of, the right whale, I think, is a baleen fish. So, that's a whole different conversation, is about the plankton that they're eating, or the krill, and then kind of how that's affected. So this is a really cool first step to see kind of how we can monitor these animals in the wild that are really hard to get anywhere near without impacting their migrations. And now we can just kind of fly overhead. Hopefully the drones aren't too loud for them overhead, and they can take those pictures from afar, track individuals, and really get a good sense for whale conservation efforts from there.
That's cool.
And we'll get a lot of amazing top-down whale pictures out of it.
Mm-hmm. Yeah, check your Google.
There'll be so many pretty pictures.
Hope they're open source. Absolutely. Yeah. Yeah, so that's the whale drones.
That's a whale of a tail.
Yes. Now, speaking of animals on film, let's say, this is a study looking at nature documentaries and whether they make a difference. So I was very interested to see this because, there's been documentaries coming out, especially now with streaming platforms. They're much more accessible than they ever have been in the past. You don't have to be watching your local PBS station at just the right time to see your nature docs, right? So you can log on anytime and you can watch a nature documentary. And so the question there is this helping animal conservation? Is it helping wildlife conservation? Is it helping with knowledge of these animals? Or is it just entertaining? Which I had my own ideas going into it, but I think it's really interesting that they did a study really to look at the impact of these documentaries. So this is an Irish study from University College Cork. And they wanted to look specifically at Planet Earth 2, which came out in 2016, narrated by David Attenborough, of course. Mm-hmm. And they wanted to track how the documentary changed attitudes towards nature by measuring audience reactions and engagement on social media. So the first thing they did is that they found that the show itself, when they studied it, had very little time allocated to conservation topics, and that those that were mentioned generated little reaction in audiences. So they're thinking, this isn't good, sounding like just entertainment, animal facts. But they found that the show generated active interest in the species it portrayed. So not just watching it for fun, but taking the next step in finding out more. And that next step sometimes lasted up to 6 months after the initial viewing of Planet Earth 2. So they looked at the species that appeared in the show, how much screen time was dedicated to the species, and what group of animals they belong to. Then they searched Twitter for 35,000 tweets with the hashtag Planet Earth 2 to see if they were reacting on Twitter. And then the last step, I love this, is that they analyzed the number of Wikipedia hits for the pages of each species. So through all this, they could assess how audiences were engaging with the individual species, with the show, and who was looking for further information. And then the very last thing they did was look for donations to charities related to those species or habitats. So as I mentioned, the interest in species lasted up to 6 months after watching the show. But they assumed that certain types of animals would have a bigger reaction than others, like mammals versus insects. But they found that the actual link to how much people looked into an individual species was all about how long that species was on screen. And this was on Twitter, how much they were talking about it, and also how much they were looking it up on Wikipedia. A really good example of that is that there's a huge scene in Planet Earth 2 where there's a swarm of locusts. And you wouldn't consider locusts to be charismatic by any means, but people searched a lot about locusts after this very extensive scene on them in the show. The effect also, love this, was independent of how well or poorly known the species was beforehand. So a lot of people know things about giraffes. They might not know a lot about racer snakes, which the Planet Earth 2 was the famous clip that most people who've spent some time on YouTube have seen, where the racer snakes actually chase iguanas, and there's a big iguana ball or a snake ball around the iguanas. It's harrowing tale.
It's one of the most traumatizing things I have ever watched on television.
Yes.
Yeah, but the thing is, racer snakes gotta eat, you know? But anyway.
Don't care.
This is their example of a species being lesser known. Racer snakes didn't even have a Wikipedia page before the show, and 2 days after that segment aired, they did. So there was an immediate spike in a desire to have that space, and then people started visiting it. The last bit of the research here was looking at donations. And as I probably could have told you, donations did not follow watching Planet Earth 2. The caveat they lead to that is that in other shows, like on Our Planet, which was on Netflix, they have a direct link to the World Wildlife Fund. And so they have this hypothesis that because Planet Earth 2 didn't have a direct link to any one conservation organization, people didn't know where to go to funnel their interest. And so if they repeated this study with that, they might see a spike. But I think also we need to remember that there are huge competitors for people's dollars and efforts in giving donations. And that, not getting a donation to a specific species or conservation topic is not a loss. If people are spending the next 6 months looking up locusts, that is a huge win in the eyes of conservation, because awareness breeds interest, interest breeds caring, caring breeds conservation and action, right? So it's all kind of part of this spectrum. And if this is how we can really see there is an impact in people's interest and exploration into a species or habitat, that's a huge win. But the last thing they kind of mentioned based on this study is that based on all of these metrics that they found and these links that they found, more time on these shows, their suggestion is, should be given to endangered species. And that's because if the amount of time you spend looking at an animal and researching them and looking at what they need, and if they need help from you, if that's going to be directly related to the amount of screen time that they get, then they need to give good screen time to endangered species. Of course, this is a whole separate thing where, like, I don't think you should be talking about endangered species. You should be talking about habitats that are endangered or are at a conservation threat. That's really the conversation we need to be having. But regardless, yes, we should be focusing our limited attention with these nature docs, since we know we're hooking people for good, is basically what this is saying.
And one thing I would point out is that also what is required is a good narrator.
Yes, David.
I feel David Attenborough. I get
forever.
The tone of the voice and a little bit of the accent and everything else and the steady pace. It's just I'm really interested. Oh, he's selling me a laundry detergent? I didn't even realize. Like, that's really that good. Because you can imagine, like, the the racer snake iguana scene being done by me, where I'm like, and the iguana is heading out looking for lunch, and oh my God, run, little iguana, run! Like, it might generate some excitement, but it might not generate an actual interest in the animals if somebody is just screaming.
So I think one of the things about David Attenborough too that is so special, if I can take a second, is that if you watch some of his original TV show where he's on camera and he's out in the wild, he's always getting down on the floor with the animals, and he gets so excited when he sees this stuff, and you can hear it in his voice too, which I think is a really big part of it, right? Is that he has a very measured tone when kind of scary things are happening, but he also is audibly excited to share this stuff with you, which
there is love
hopefully
in
that comes through on the show as well.
Absolutely. I hope so. I do hope so. Yeah, and this is I find this really interesting, but it does get at, a point of fundraising for so many organizations. It is about the ask, and it is about telling people where to go and how to help. And if the goal of these nature programs is to help endangered species, is to help conservation efforts, then the show needs to be aligned with certain things that people can do.
Yeah, you got to have that next step, right?
You have to give people something to do.
That's a good kind of tenet of education, right? Never present a problem without a solution.
And an easy way to do that is something like through a Netflix or something that is online content where you can actually have a button. The thing that I thought of as soon as you mentioned the Netflix, I thought of the zoo. I don't know if your zoo does it. You get the little metal token and you can drop it in.
Oh, yeah.
They give you a selection of 1 or 3 animals that might be endangered or need fundraising. And you drop your little token in there, and then supposedly somewhere down the line they get for whoever collects the most.
They get a giant check, like a prop.
The animal gets a
they hand
prize.
it to a zebra, right? Yeah. Absolutely.
So, but you could have a very easy tie-in where, you're talking about this iguana or what have you, and have a little $1 donation thing to protect that they pop up, and you can. Click it and it comes right out of the Netflix account on the next bill or something simple that way that not only makes it ties the connection to there needs to be help and go look at this later and 6 months from now, hopefully you've done it. But while you're looking at this, here's a thing you can do right there in the moment.
And I really do.
Netflix, are you listening?
That's what I was going to say. I hope that Amazon, Netflix, the platforms that are streaming these programs, that they can incorporate that. Are you listening?
Absolutely, yeah. And I will just mention in the chat room, just because it makes me feel very warm and fuzzy, Noodles did mention, I looked into polar bears after last week's show. So there we go. That's what it's all about.
The effect, Blair. You got the Edinburgh effect going on over there.
Oh my goodness. A dream come true.
Well, a dream come true for me is to get to the break. No, I'm really enjoying the show. And
I'm about to lose power. I got to go plug in.
Oh, you go plug in. I'm going to take us to the break. It is time for the break right now. Stay tuned, everybody, for a bit more of This Week in Science coming up in just a few moments. Thank you for sharing your time with us and for being a part of the This Week in Science community. Did you know that our calendars are available for pre-order? That's right. I have been trying to let you all know that our calendars are available and you can get yourself on the list to get a 2020 Blair's Animal Corner calendar from TWIS in time for the holidays. One for you, one for a friend. Sounds about right. Blair has made a stained glass effect this year for her art, and we are excited about all of the pieces of art that will be in the calendar this year. So go to twis.org And click on the, what is it a hypnotoad, Blair? The hypnotoad?
It is a horned frog.
Oh, sorry, the horned
It's a
frog.
real species, actually. Yeah.
Okay, the cover of the calendar, the horned frog, as it has been for the last 4 years?
Well, my first one was 2016. So 16, 17, 18, 19. This is the 5th anniversary.
Fifth
this is
anniversary
the 5th year.
calendar. There we go.
Uh-huh.
You can pre-order yours now over at TWIS.org. That's so exciting. And while you're there, if you have not subscribed to TWIS, you can click on the orange subscribe button, and that will open up your options to YouTube, iTunes, Google Play. You can send other people over to TWIS.org to be able to subscribe easily in that manner. Or you can go to whatever location you like best for listening to your podcasts. Additionally, if you are interested in helping to support our financial needs for running the show, you can head over to Patreon. Patreon, we're having a limited time offer right now, only until the 14th. So we are just about 5 days away from the end of our special offer. If you sign up to become a patron, hit that orange red button over at patreon.com/thisweekinscience. Sign up at the $50 a month level. You will be eligible to get a piece of original art by Blair, some of the calendar corner art, the original pieces framed. And that's when you support at that level for 3 months. So if you support for 3 months, we will send you a framed original piece of artwork by Blair. And that is only until October 14th. So if you're already at that level, you're in. If you're not at that level yet, you can get in there right now and become a patron at the $50 a month level. Get yourself a piece of framed art. Know that you are supporting us as we head into the end of the year, and that you're helping us to make this show happen and get the things done that we need to get done. So Patreon, you can click on the Patreon link at twis.org, or you can go directly to patreon.com/thisweekinscience. Thank you all for all that you do to help support TWIS and to keep it going. We are listener supported at this point, and we love to be beholden to you. So thank you for all that you do for TWIS. We really could not do any of it without you.
And we're back with more This Week in Science.
Oh yeah, we are back again. Now it's time for This Week in What Has Science Done For Me Lately. Our letter this week is from Tom Merritt. You recognize that name?
That sounds familiar.
Yeah, host of the Daily Tech News Show.
Friend of the show.
Friend of the show, for sure. Tom writes in, says, what has science done for me lately? Science has granted me power. He didn't put the laugh in. No. He goes on to say, solar power, to be exact, thanks to advances in photovoltaic materials and, of course, the sun. I'm now feeding energy to the grid during the day and getting credit for the power I pull back out at night. Not only is this good for my power bill of, if it all goes well, it should be about 0 for electricity on average. That sounds really good. Yeah, but during the day when air conditioning is straining the grid, I'm helping contribute, not just adding to the load, not to mention lowering emissions a little bit. That's good. So thanks, science, for figuring out how to make photons into electrons in a way I can afford to do on top of my house. Thanks, science, for sure. Thank you, Tom, for writing in. Thank you for sharing your This Week in What Has Science Done For You lately. I know there are many other people who maybe are using solar power in California as the electrical grid is being turned off. You have some any solar panels not connected to the grid that you can use to power things in your house, maybe? Yeah, California, you're under some things right now. But solar, I mean, if we can all pitch in whatever way we can, it's amazing. And solar is getting more and more efficient. The science is, it's mind-boggling. It's wonderful what we are able to do now. And one issue in urban heat islands is that of dark-colored roofs causing increases in heating. And if that dark color is not just absorbing heat onto a roof tile, but into a solar panel, this light of the sun producing
both,
electricity,
actually.
or both. It
Or
could
both.
be great. Heating water. That's right.
I actually saw a building that I had to do a second take. At first, it looked like just an architectural design. The tile roof was sparkling. And I went, oh, that's kind of cool. Put a little glimmer, and then I realized, oh no, that's tiled solar panels on this roof. This is they're capturing energy for this building through the roof, and it's still in tile form, so it still has the fit in sort of aesthetic with the buildings around it, except you can kind of tell because there's that little bit of sparkle.
It's like a unicorn house. It's great.
It's a big unicorn house. But yeah,
I like the solar ivy too. The solar ivy
sparkle
so
pony.
Pretty. Yeah.
I want a solar sparkle pony house. That would be great. Okay.
And this was a large building too. This was like maybe a 4 or 5 story building.
Wow.
Where the entire rooftop of this
That's cool.
building.
Very cool.
So, yeah.
Thanks, science. Thanks, Tom. And if you have something you want to share with us, write in. Let me know. So you can send me an email at Kirsten, KIRSTEN, at thisweekinscience.com, or leave a message on our Facebook page. That's facebook.com/thisweekinscience. Just leave a message and help us keep filling this segment of the show with your stories and your notes and your poems? No, your songs and your poems. And anyway, it's time for some more science. Justin.
Oh.
Tell me a cool story.
All right, what do we got here? Next up, this is I goofed up. I said previously I started to say that microRNAs were involved in the antibiotic. That one was working on ribosome. This is one that's the ribosome. Thank you. This is one that is actually has the microRNAs in it, and it's cartilage in human joints. They're finding repair themselves through a process that's not unlike the regeneration that you see in salamanders and zebrafish that use this to regenerate limbs. And part of this is sort of interesting. And there was something that had been noticed medically over the years of treating humans, which is that when there is an ankle injury, a knee injury or hip injury, ankle injury seems to recover much quicker than the knee injury, which recovers quicker than a hip injury. Hip injuries to cartilage can become arthritic at higher rates than the knee, and the knee can become at higher rates than the ankle. And what's sort of interesting is the process that of this cartilage sort of repair is occurring with greater affect the further away from the core of the body is, the outer limbs, which is also it's also what happens with like a salamander tail or the outer extremities on the zebrafish. It's sort of like the furthest tips have greater regenerative ability, and this is researchers have further learned that microRNA regulate the process, and that these microRNAs are more active in animals that are known for limb, fin, or tail repair, which adds back to the salamander, zebrafish, and a number of lizards and this sort of thing. These microRNAs that they discovered are part of the process of regeneration are also found in humans, and it is, they think, an evolutionary artifact that is providing the capability for human joint tissue repair. And it's one that you could quickly imagine why this would be conserved in humans, or maybe pulled out of the dustbin a bit because humans for so long did so much running that cartilage repair would be absolutely something, a trait that would, through natural selection, be very beneficial for what humans were going to survive.
I want to know. I know so many people with knee and hip injuries, I mean, part of it is age. These knee and hip injuries tend to start popping up in your post 40s. These injuries start being deleterious. And maybe, that's past the age of survival early in our evolution. But yeah, I've always, even as a physiologist, I know that cartilage it's all bone and cartilage. This is all living tissue. But at the same time, cartilage is the thing, does not repair. It doesn't have blood vessels. It doesn't, anybody who's had the cartilaginous part of their ear pierced will know that there's still a hole going through your ear because the cartilage doesn't fix itself. If the cartilage in your knee gets messed up, you need to have knee surgery. It doesn't fix.
So, it turns out it does, and
that's what's fascinating me about this.
But and the interesting thing, you know so many people with hip and knee injuries, but ankles. How many people are like, I had the cartilage in my ankle has gone bad, so I can't. It's because of this discovery is answering the question as to why it is that you don't hear so much about debilitating ankle injuries at the same rates, right? So and those injuries may be just as common, but it's a sprain and you wrap it and then you stay off your foot for a couple of weeks and you're fine. So they're calling it our inner salamander capacity. And they're excited to learn what's regulating the regeneration and the correlation to what's taking place in salamanders and humans. This is quotey voice from one of the researchers, Krauss, who says, we believe we could boost these regulators to fully regenerate degenerated cartilage in an arthritic joint. And if we can figure out what the regulators are missing compared to salamanders, we might even be able to add the missing components back and develop a way someday to generate part or all of an injured human limb. We believe this is a fundamental mechanism of repair that could be applied to many tissues, not just cartilage. So that, Blair, if you're going to live forever, you're going to need healthy hip, knees, and ankles. And occasionally may need a new limb.
By all means, sign me up. I could use a new right hand at this point. It's still giving me trouble. I drop things all the time. Yeah. But I'm very dubious. I'm very it sounds too much for me. That sounds very Harry Potter. Like Skele-Grow, time to grow some new bones. I don't think so.
It's just I do think so because we do
the world of animals though.
Yeah, we see it in reptiles. We see limbs regrown. Salamanders, as the key example here. Right. Tails regrown. Lizards lose their tails all the time so that they can escape a predator, and then
but they don't get their bones back.
Okay, but what we're talking about is cartilage.
Yeah, but no, it's
which is structural.
The extra jump kind of to other things. I think the cartilage makes sense to me. Absolutely. Your ears keep growing as you age, even when you stop growing, right? So there's other biological processes where that's not too hard for me to believe, but the extra jump is much harder for me. And not to say that it's impossible, but it is much harder for me to get there.
And you're right. It may require a bone grafting thing of some sort to create the skeletal framework. But there are, how much more complicated can you get than an eye? And there are, is it fish and frogs that can do this? I'm not sure.
Mm-hmm.
I remember the species, but there are at least fish that can regenerate an eye that's been damaged. So when you're talking about what we're talking about here is cartilage, because that's what's being regenerated in this case, but it's not the only thing that can and in nature undergo this. So yeah, it would take some sciencing to make it so it can be
but the
translated
basis
into other.
Yeah, the basis of this is not just cartilage, though. The basis of this are these microRNAs, which are these, which go around and shut things off and turn things on. And if you have these little control molecules that you figure out which ones control different aspects of cellular division and protein translation, then you can go in and give some instructions to a damaged area of the body, instead of maybe, only doing, what an idea now is stem cell transplants for the knee, right? If you have a damaged knee, you can put stem cells in there. The stem cells don't necessarily know what to do, but then if you know which microRNA to turn on at the same time, potentially you can create an environment that is conducive to the stem cells going, now I know what to do, and helping the cartilage grow, or even just upregulating the cartilage fixing itself. And this is turning on light switches. It's turning things, opening up the floodgates. It's turning it on, turning it off.
Well,
if you know how to control it.
But what it's
works.
turning on and off, I think, is also interesting, because then we're talking about conserved DNA that we may not be utilizing. Right. And now you've got something that can turn that switch on. And then you can also then look and see, okay, well, maybe the machinery that's behind it isn't there. And so then you're talking about genetic modification or creating a pathway outside of the genetics to start that process of regeneration. So you can either simulate what DNA has already been doing, or you may be able to turn on that sort of latent DNA, that junk DNA that's been hanging around since fish.
Not since fish, hanging around since we started growing, and then our bodies turned it off. Because so much of this absolutely. To regrow a limb, our body already did that once, and then it turned all those genes off. It's just turn those genes back on again.
We've done that before.
But would it take 20 years to grow?
No. Yeah. You'd regrow your hand, Blair. But it would be this cute little baby.
Oh my god. Of course, also now I'm picturing in trials that they try to grow just some hip cartilage and they end up with a third leg. Oh no, we flipped the wrong switch.
Hey, if you can run with it, run with it. It might be quicker.
I think it'd be strictly ornamental.
You might win that 3-legged race yet.
After
all. Moving on to some other crazy, awesome things that scientists are doing. Researchers have used optogenetics to implant a false memory into the brains of birds.
Oh, okay.
Yep. He said, hey, birds, you didn't really do this. You didn't really learn this, but now you know this.
There was something like this with rats that we did just a few months ago, I think, right? Does that sound familiar?
So it does a little, yes. And the optogenetics possibly, yes. Researchers in this particular study, which was published in Science, were looking at zebra finches, which are a model animal, my favorite from grad school, that is often used to study birdsong. The zebrafinch song is great for the study because it's a very stereotyped song, but the babies learn the song from their fathers, and they go through a period of listening to the father sing the song. Doodle-loop, doodle-loop, doodle-loop. That's my little zebrafinch impersonation. And then, so after the listening takes place, the offspring, the male offspring, will then practice, and they'll go, and they'll make a bunch of mistakes, and then they'll start to crystallize on the adult song, which is close to the copy of their parent song. There's a lot of stuff that's innate in there as well because it's stereotyped. The learning happens, but there's also a pattern in the bird's brain that allows the practice, this plastic part of the practice phase, for a comparing within the brain. So the offspring knows I'm not doing this right and can pattern match to create the final song. And we know that there are very specific areas of the brain in the bird brain that are involved in creating this whole process. And researchers have been studying this for a very long time. And there are several areas of the brain researchers have been looking at for a while. One of them is the HVC, or the higher vocal center, which is for the production of the song. And so a lot of areas of the brain in this whole process, researchers think mirror a lot of the wiring in the human brain and our own learning of speech and language and the production of language as well. And when birds are learning a song, their auditory cortex contains a region called the NIF, the nucleus interfacialis of the nidopallium, and the NIF connects to the HVC. So you have the heard song that then puts input into the motor production part of the system, allowing the animals to produce something similar to what they heard. Okay, this is where it gets really interesting. The researchers kind of took things over. So the researchers in this part of the study, they used optogenetics to bypass the actual listening part. They used light to trigger neurons in the NIF, that little area of the brain that sends information to the HVC. And they did so in a way that they would leave the light on as if that was a note being sung by the tutor. So the longer the light would stay on, that's the longer the sound that the young bird would want to produce later. And so in effect, they couldn't actually create a difference in the sounds. So there's no melody and not a lot of variation, but they could create a pattern, a rhythm for this song. So it would be kind of, instead of a bird singing doodle-doot, doodle-doot, they would sing doot, 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 all the same sound. But they showed that they could trigger the learning to take place. Birds that received the optogenetic light stimulation in the NIF area of their brain sang messed up songs that they thought they learned from a tutor, but they actually never did. They never heard a single song ever.
Wow.
And birds that they did not get that kind of training didn't sing at all. They didn't get a song.
Yeah. That is wild.
So I found a study, I don't know if we talked about this on the show, but it's sounding so familiar to me, from May of this year, where using optogenetics, they were able to kind of insert a memory into mice of a specific smell. Yes. So it's definitely, it's getting more complex, it sounds like.
It is. What I think is fascinating here is this. We are really starting to figure out the data processing part. I mean, there's still so much to learn, obviously, but we're learning this data input to processing to output aspect of the brain. And so what are the signals that a certain area of the brain needs to receive to make something happen? And it's this idea of the brain doesn't know what is real and what is fake. It just knows input. It's like Johnny Five from Short Circuit. Mm-hmm. More input. That's our brain.
Yeah. So it's something that's, I think, taken for granted nowadays, but you can go into an audio editing type program generate a sound, manipulate that sound. I've seen people who turn these into beats and tonals, so they can write music from scratch on a computer. Which we sort of take for granted. That's how things are done. But there was, for most of playback history, at least recordable history, you had to have a sound go in first to put a sound out. And so what we are sort of, like you said, tapping into the data generation mechanics of the brain, where you don't actually need specifically a sound input to have a sound output memory that can be played back, which is, we're starting to decode brains in general. Which is that a good thing? Is there a frightening application to this? Do we want brains to be programmable? Yes, we do. Yes, we do. We absolutely need people to have programmable brains, if for no reason other than to
create a dystopian future, of course. Yes.
Yes.
No.
That's how you start. You have to start with the human brain. Yeah.
So I just want to let you know that the goal of this study, of the eventual goal, is not to program people's brains with false memories, although, it's kind of starting to look that way.
You can do that from the outside already.
You can. Exactly. Yeah. But the idea is that this is going to help us pinpoint more specifically genes and neurons that are involved in language production, and that because of the similarities between the bird brain and the human brain, that we will be able to help human individuals who have conditions that affect their ability to vocalize. So that's,
interesting.
That's the eventual goal, is to understand the circuitry so that we can pinpoint it and fix it.
Yeah. The first thing I honestly did think of, though, was there's forms of schizophrenia where people have audio hallucinations. So this is audio that's being generated from within the brain and not external. Yeah. So there's potential therapeutic applications.
Probably.
Understanding how these things could be being generated.
Yep. So it's pretty cool stuff. This is cutting edge research, putting memories into, as Todd Roberts, a neuroscientist at the University of Texas Southwestern O'Donnell Brain Institute, said, "This is the first time we have confirmed brain regions that encode behavioral goal memories, those memories that guide us when we want to imitate anything from speech to learning the piano. And the findings enabled us to implant these memories into the birds and guide the learning of their song." Waha. Justin, tell me another story.
So I have a discovery of a new family, genus, and species of microinvertebrate that doesn't have an extant group of fossils that it fits into, and as of yet, they can't figure out if it has a current existing relative either. This is a 30 million year old fossil that was discovered in amber. The findings are by George Poinar Jr. of the Oregon State University College of Science. And he's informally calling the new animal mold pigs.
Adorable.
Because they have a resemblance, I guess, to swine. Actually, to me, it looks very much like a four-legged tardigrade.
Yeah, they look like a tardigrade. Yeah, and a moldy tardigrade at that. Yeah.
Yeah. So the way they got the name is pretty interesting. So it says here too. Pointer says occasionally, as in the present case, a fragment of the original habitat from millions of years ago is preserved. The mold pigs can't be placed into the group of currently existing invertebrates. So they don't have the extent. Fossil relative either. They share characteristics with both tardigrades and mites, but clearly belong to neither group. So they've had it's an interesting find because they didn't just find a one-off here. They found several hundred individuals preserved in amber, and they can see their sort of warm, moist surroundings with pseudoscorpions, nematodes, fungi, and protozoa. So you actually have the ecosystem that they were living in. And because of the large number of fossils provided, additional evidence of their biology, including reproductive behavior, development stages, and their food sources, also could be sussed from this. So this is really interesting. The discovery shows that this unique lineages were surviving 35-ish million years ago. Right? And it's sort of a snapshot, and it's, they don't know. They don't know the history, and they don't know its future. They think it fed mainly on fungi and supplementing that food source with other small invertebrates. But the thing that's sort of separating this from the mites and the tardigrades is that they don't have claws at the end of their legs, for one thing.
Mm-hmm. So they can't grab onto things as well.
Yeah. So it's a really interesting snapshot, an alien of sorts on our planet, and that we don't know exactly how it fits into this family tree. It's its own genus, family, and species all in one there. It's just
so. Can I speak to that for a second? Because as the resident who believes that tardigrades are aliens, and Justin, you might be with me on that one. This is kind of bursting my bubble a little bit. That these guys are here. And they're similar enough. They're filling a similar niche, and they're here. They were here. It makes tardigrades fit in the evolutionary timeline a little bit better, because if there was this radiation of things like this living in water droplets doing these things, then the tardigrade isn't so weird. And that makes me sad, but it does ground me a little bit better and make me think that, yeah, all right, maybe tardigrades are ours, but maybe they're not. Maybe they're both from outer space.
Where
but I feel like they're probably both ours.
Of course, but if you did want to hang on to your transpermia tardigrade origin story. You can say, obviously that planet had an abundance of tiny invertebrates, and we got more than one.
They were that planet's beetle.
It was just everywhere.
I prefer the idea that maybe, even though panspermia, that maybe things started here, and tardigrades from Earth are spreading through the universe.
Mm-hmm. Or moss pigs. Maybe some moss pigs got out before they went extinct.
Mold pigs.
Now we have moss pigs and mold pigs. Yes.
Moss pigs and mold pigs.
There you go.
Yes.
Oh, it's good to have a veritable cornucopia of pigs.
An abundance of pigs.
And let's talk about special striped cows now.
Oh, please, let's.
Oh, please, let's. Yes. Researchers, spurred on by so many studies that we have talked about on the show that, Blair, you have brought during the Animal Corner about zebras, stripes, and what are they good for?
Absolutely nothing. No.
Except maybe avoiding those biting fly attacks. Well, the researchers looked at the research and they said, we've got to put it on a different living animal and see if it works. And so they painted some cows.
Great. Love it.
Yes, they painted cows in black and white stripes all over the body. And they just, not a huge sample size, but then they checked to see what how the animals fared in terms of insect bites. And lo and behold, the stripy cows were bitten less and also acted like they were bitten less. The cows didn't have the stress behaviors or the bite avoidance behaviors to the same degree as non-striped cows. Wow. And so this is further evidence that painting a cow like a zebra could potentially keep it from being bitten by biting flies, which in some areas, because of the amount of biting flies, can actually have a very detrimental effect on the agricultural revenue from those cow herds. So,
And so we've got a
they're
picture of this.
they're recommending that maybe the cow farmers should paint their cows.
Okay. So,
What about
so
genetically engineered cows to be striped?
They could do that as well.
We've got a picture of this cow, and they didn't just put on some stripes. They really went to the extra effort to kind of try to recreate a zebra pattern here. This is a pretty well done cow dressed up like a zebra.
It's like Halloween came early to Cowland.
Yeah.
Yeah. This would definitely cause a double take driving by the pasture.
So I think this is interesting because we had a study where people put horse coats on some horses that were zebra striped, but in this case, some of the worst bites that can actually seriously hurt these cows are on their legs, which is probably why they just went for it and painted them.
Yeah.
Yeah. They painted them all over. So yeah, it was good. And they suggest that this would be a way that it's an alternative method of defending cattle against biting fly attacks
And it's
without
a fun,
using pesticides, right?
Yeah.
So yeah.
It's a fun weekend project for the farm kids.
Mm-hmm.
What are we doing today? We're going to go paint some cows.
Here's some whitewash. Go paint your cows.
So
In
in
Nope,
colder
break out.
areas, perhaps cow sweaters are going to be a thing that are zebra striped. But otherwise, I would say, yeah, some genetic engineering to make them striped would be pretty good. But I'm also hearing that when I go into biting insect territory, I should be wearing stripes, which I had not really thought about.
You might want to look like a zebra.
Yeah.
Mm-hmm. Everyone, it's going to be the new field clothing line. Zebra skin, but not
Love
really
it.
zebra skin. Zebra stripes. Yes. I've got a few quick stories for the end of the show. So this one I absolutely love because it just puts us in our place in the solar system. Voyager 1, as we know, is outside our solar system, right? It is past the edge of what we consider our solar system, headed out toward the Oort cloud, toward the heliopause, and the past the termination shock. And Voyager 2 is well on its way in this endeavor as well. Researchers wanted to know what kind of pressure, what kind of forces are out there at the edge of our solar system, at that boundary between our solar system and the rest of space, interstellar space, right? So here on Earth, gravity, or not gravity, but here on Earth, we have here on Earth, air pressure. We know there's a bunch of molecules and they weigh down on us, and down here at sea level, it is a certain amount of pressure, and up at the top of a mountain, it is a different amount of pressure, less pressure, because there are fewer molecules pushing down on your head. Here at the surface of the Earth, it's 1 atmosphere of pressure, because you got the whole atmosphere on top of you. When you go down beneath the ocean, you have more molecules pushing on you, greater pressure. But as we leave the solar system, you're leaving behind the particles that are contained in the solar wind. They push outward, but as they go ever outward, it becomes fewer and fewer. So that pressure you would expect to be a bit less. And so researchers were using the Voyager spacecraft to figure out what was happening in the areas far away from the center of the solar system. And they used an event called a global merged interaction region. Say that 3 times. Global merged interaction region. This is caused by activity on the sun. Solar flares. The solar flares fire out and they release bursts of particles, right? These are, so solar flares, otherwise known as CMEs or coronal mass ejections. They eject a bunch of particles into space at very high velocities. And as they travel out into space, they kind of create a wave, like a bow wave, if you can think of it that way, creating a wave of plasma that gets pushed by magnetic waves. One wave like this reached the heliosheath in 2012, and Voyager 2 recorded what happened when that happened. And then a few months later, scientists saw a similar event as a result of observations in Voyager 1. And in both of these, what they saw is that the wave caused the number of galactic, so stuff coming in, galactic cosmic rays, to decrease. So it was this pressure, this wave front from the sun pushed galactic cosmic waves away, repelled them a bit. So it increased those coronal mass ejections that spread out particles from the sun that actually repel interstellar space particles. So they were able to take the distance between the spacecraft to calculate the pressure in the heliosheath and the speed of sound. So trivia for everyone, heliosheath speed of sound is 300 kilometers per second. It's much faster than it moves through air, so not much out there. They also noted that the change in galactic cosmic rays wasn't the same at both spacecraft. Voyager 2 had a different decrease than Voyager 1. Voyager 2 was still inside the solar system, and Voyager 1 was outside the solar system. And what they found at Voyager 1 is that only galactic cosmic rays traveling perpendicularly to our solar system's magnetic field decreased. And so there's an asymmetry, and so something happens. Researchers don't know what happens yet, but something happens as the wave transmits across the heliosheath, the boundary of our solar system. Something is different in the way that cosmic rays interact inside the heliosheath and outside the heliosheath with our sun's energy. So, pressure at the edge of our solar system. And all I can just think of is just how we're like in a bubble. Our sun is creating this bubble that's zooming around space, and it's pretty awesome. What's going on outside that bubble? And then my last story for the evening is a story about new moons. If you've heard, Saturn got 20 new moons. Brings it up to a total of, 82 moons. Go Saturn. The majority of these are really small, but now it apparently has more moons officially than Jupiter. So it now is the top moon haver in our solar system. Researchers who have been working on this think that Saturn may have as many as 100 moons, but they're like little pieces of dust. The bits that are around are probably under a kilometer in size, so they're very hard to identify from where we are. But this means also that these 20 new moons need names, and there is a contest from the Carnegie Institution for Science to help name the new moons. They must be named as the other moons of Saturn have been named. They must come from Inuit, Norse, or Gallic mythology. But you too could be a part of naming the 20 new moons of Saturn. We will have a link at our website.
I'm glad that they put a caveat on it. So we don't end up with a Mooney the moon face.
Yeah.
I was thinking the same thing. What do you mean?
That was a home run the one time, and now it's just...
Yeah, that's not Norse
so
mythology.
cliche.
No. All right. Blair, what's in bird pee?
Oh my goodness. Well, first of all, I have to real quick explain for all of you that don't know that birds don't pee. They don't have separate exit points for different types of waste. They have a cloaca, which I like to consider the Swiss Army knife of orifices. They do everything. And so out of it comes kind of this mixture of pee and poo. The conventional knowledge is that urea is what's in mammal pee, and uric acid is what's in birds, reptiles, and insect pee. And so this kind of differentiation, is in trouble. A recent piece of research out of University of Texas at Austin looking at birds and bird excretions from the Austin Zoo have found no sign of uric acid in any samples.
No sign.
Interesting.
The analysis found ammonium urate and struvite and 2 other unknown compounds, but absolutely no uric acid. The current hypothesis is that there is some sort of bacteria inside bird guts that break down the uric acid before excretion, but we are not sure. So everything that you think about bird poop and pee, like the reason that a bird poo on your car is really hard to clean off and might eat away at your paint, you always say it's the uric acid. But that might not be the case. So
How are we only just now just looking at that?
well,
This
it's
is what I
interesting
wanna
you
know.
bring that up
What
because
happened?
actually there was someone named Bob Folk who in the 1960s posited this. He claimed that bird waste did not contain uric acid, and generally speaking, he was laughed out of lecture halls, and he was kind of told he didn't know what the heck he was talking about. He found no sign of uric acid in samples collected in 17 species in the 1960s. And so that paper was published in 1969. It used X-ray diffraction and solubility tests and found nothing. But so this newer study was using some newer technologies, some of the same technologies, and looked at a pretty wide array of types of birds in the Austin Zoo and just found nothing.
Wow.
So more to look for
Goodness.
there.
I love this though. The story here, says that the researcher, one of the researchers, Julia Clark, she had a conversation with Bob Folk, who claimed that the waste didn't contain uric acid. And she says, sometimes you just get presented with really weird question and you want to know the answer.
Yeah.
There you go.
So the first
I'm glad
thing,
she looked into it.
well, I'm sure the first thing she looked for was any paper anywhere that showed it being found. And then that's a kind of an interesting null hit if you go and look for, well, obviously there's a counter argument to what this person is saying. And you go and look and you're like, I should be able to find this. There should be a paper. There should be research done. There should be somebody talking about utilizing or effect. And if you don't find anything, that's a big aha. Okay, now we have to test. Now we have to do the look because you can't find it anywhere in the body of research.
It's just so interesting because this was a test question in so many classes in college.
So many.
What is the waste product in bird waste? Uric acid.
Yep.
That's what it is.
I know for a fact that human pee is urea, and I know this because of physiology lab, and it's fine. This is not a question.
And this isn't saying that also perhaps, reptiles, amphibians, insects, that's not found in there. This is only saying for sure what they know about birds right now, which is that birds appear to not have it. And the explanation
just opens up new questions.
Is not that it was never there, but that there's some sort of process happening that's breaking it down, is the current hypothesis. So lots of unanswered questions.
It's going to lead to new research.
And the one I want answered first is, how did this get entered into the body of knowledge? in the
Where
first
did it
place?
come from?
Great
Mhm.
question.
That's the first thing. It's like, how did that start to the point where it could be a test question, and yet was itself never tested?
Yeah.
Mm-hmm.
Tell me a story about canned soup.
Yeah, well, this is Tel Aviv University researchers in collaboration with scholars from Spain have uncovered evidence of a 200,000 to 400,000 year old canned goods in the Kesem Cave near Tel Aviv. By canned goods, of course, we're talking about the storage and delayed consumption of animal bone marrow.
You know, Tupperware. It was the Tupperware of the day.
Tupperware of the day.
Yeah.
So these
I got a
early
bone here.
Paleolithic
Yeah.
people would save animal bones, they think, for up to about 9 weeks before
Whoa.
feasting on them in the cave. So they got all these bones. They've got all of them in sort of these interesting states with skin still on them. Researchers evaluated the preservation of bone marrow using an experimental series on deer. They controlled exposure time, environmental parameters, combined with some chemical analysis. A combination of archaeological and experimental results allowed them to isolate the specific marks linked to dry skin removal and determine a low rate of marrow fat degeneration of up to 9 weeks of exposure. This is quoting voice of Dr. Belasco. We discovered that preserving the bone along with the skin for a period that could last for many weeks enabled early humans to break bone when necessary and eat still nutritious marrow inside. The bones were used as cans that preserved the bone marrow for a long period of time until the skin was taken off. Shatter the bone, eat the marrow, says Professor Barkai. So this is in the October 9th issue of Science Advances. And it's kind of an interesting story because we have been conceiving of the hunter-gatherers as needing to eat at the site of the kill almost. You know, like this is the environmental pressure on these hunters is that you always need to be killing something in order to eat. But this shows that they were actually already starting to figure out food preservation techniques. Excuse me. What's also interesting about this cave is they don't know who these people were. They don't have apparently any human remains to say who this. So this could be a Homo erectus. This could be a Neanderthal. This could be early humans, sort of getting out ahead of the out of Africa migrations or one of the earlier events of. But they as of yet don't know who these people whose artifacts they've been sifting through are.
It's old though. This 420 to 200,000 years ago. So that's old.
It's
Yeah.
It's very old. So that, but it's, and then where it is though is sort of in the corridor of humans leaving Africa. Or the current modern humans as we are defining them. And is also areas that were inhabited by Neanderthals and Homo erectus. So there's just so many dang humans running around the world at that point in time. And in that location is sort of the nexus of a bunch of it. So yeah, don't know who they were. But one day maybe we'll find out.
I hope that we're able to find out. That would be pretty interesting.
But it just checks off another thing on the list. Fire, no, that was around for a long time. Tool work, no. Glues, no. We had adhesives predate current modern humans. Oh, preservation of food. There you go.
No, but yes, there we go. We have it, and we have done it. We have come to the end of another show.
Oh, wow.
Yeah. Wow. I would love to say thank you to everyone out there. Thank you for watching. Thank you for listening. Thank you to our chat room. Thank you to Gord McLeod for monitoring our main chat room. Thank you to Fada for helping out over on YouTube and with our show descriptions on YouTube and our social media. That's huge. Identity 4, thank you for recording the show so that we have an audio podcast. And thank you to our Patreon sponsors. Thank you, Paul Disney, Ed Dyer, Andrew Swanson, Craig Landon, Andy Groh, Ed Stu Pollock, Philip Shane, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Richard Porter, Mark Mazzaros, Jack, Matthew Litwin, Jason Roberts, Bill K, Bob Calder, Time Jumper 319, Eric Knapp, Richard, Brian Condren, Dave Neighbor, Gome K, Donald Mundis, Sarah Forfar, Dan K, Matt Bass, Darwin Hannon, Patrick Pecoraro, Ben Bignell, Gene Tellier, John Gridley, Corinne Benton, Adam LaJoy, Sarah Chavis, Rodney, Tiffany Boyd, John Bertram, Mountain Sloth, Seth O'Gradney, Stephen Albaran, John Ratnaswamy, Dave Friedel, Daryl Myshak, Paul Ronovich, Sue Doster, Dave Wilkinson, Noodles, Kevin Reardon, Christoph Zucknerek, Ashish Pants, Ulysses Adkins, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Suzuki, Jim Drapeau, Greg Riley, Sean Lamb, Steve Leisman, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert, Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Lucen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, and EO. Thank you for all of your support on Patreon. And if you are interested in supporting us, you can find information at patreon.com/thisweekinscience, or just click that Patreon link on our website. We are now running a limited time offer until October 14th. So act now, become a patron at the $50 a month level. On next week's show, once again, we will be back broadcasting live online with all the science that you love this show for at 8 p.m. Pacific time on Wednesday evening. You can watch it live and join our chat room at twist.org/live. But don't worry if you can't make it, because you can always find those past episodes at youtube.com/thisweekinscience or twist.org.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science anywhere good podcasts are found. If you did enjoy the show, remember to tell your friends about TWIS.
Oh my God. For more information on anything you've heard here today, show notes will be available on our website. What's our website? I'm so glad you asked. It's www.twist.org. While you're there, you can also make comments and start conversations with the hosts and other listeners. And while you're there, hey, maybe go ahead and just pre-order some of those 2020 Blair's Animal Corner TWIS calendars.
Yes, or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in the subject line. Otherwise, what will happen to your email?
Your email will be spam filtered into oblivion.
You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes tonight, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from this show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And This Week in Science is coming your way. So everybody listen to everything we say, and if you use breath instead of rolling a die. We may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.